In this lesson, we will examine concentration cells, a special class of electrochemical cells that produce electrical energy solely from a concentration difference, not from different substances. You will learn how they work, how to predict electron flow, and the conditions that drive them toward equilibrium. What is a concentration cell? A concentration cell is a galvanic voltaic cell in which both half cells contain the same species but at different concentrations. The electrodes are usually identical. The spontaneous redox reaction occurs because a concentration gradient exists, not because of a difference in substances. Key idea: the cell will generate a voltage. The potential of the cell is larger than zero until the concentrations in both half cells become equal. How does a concentration cell work? In a typical metal ion concentration cell, both electrodes are the same metal, example zinc. One half cell contains a low concentration of zinc 2+, and the other contains a high concentration of zinc 2+. Electron flow direction: electrons flow from the electrode in the lower ion concentration where oxidation occurs to the one in the higher ion concentration where reduction occurs. Why? The cell moves toward equilibrium by oxidizing metal atoms into ions where ion concentration is lower and reducing ions to metal where ion concentration is higher. Predicting the spontaneity and direction: the driving force of a concentration cell is the tendency to equalize ion concentrations, which results in a spontaneous redox reaction. Although the standard cell potential, E0, is zero, since both electrodes are the same, there is a non-zero actual cell potential when a concentration difference exists. This potential can be estimated qualitatively. Greater concentration difference gives greater potential. As concentrations equalize, the potential of the cell decreases towards zero. Example: a concentration cell is built using two Ag electrodes. One half cell contains Ag plus concentration equal 1 molar. The other contains Ag plus concentration of 0.001 molar. The electrode in 0.001 molar Ag plus will act as the anode, oxidation. The electrode in 1 molar Ag plus will act as the cathode, reduction. Electrons will flow from the dilute side to the concentrated side. Outcome: the cell spontaneously generates a small voltage as the system works to equalize the concentration of Ag plus in both compartments. Summary points: same metal electrodes, different ion concentrations. Spontaneous cell driven by concentration difference. The potential of the cell is non-zero only when concentrations are unequal. Electrons flow from low to high ion concentration side. If you found this helpful, this was just a small part of a full in-depth chemistry course I created. In the full course, you will find structured lessons, downloadable resources, practice problems, and quizzes to really master the material. Check the link in the description to start learning the full course today. I would love to see you inside.